Hey guys, we're getting there, man. We're almost there. We actually have been jumping ahead and looked. We only have about 2 to 3 more major ideas in ecology, and we are done. It's how much you actually know. I know it doesn't feel like we know a lot. You know a lot. So today we're gonna go back. I know it's gonna seem kind of crazy. We're gonna go back again to Mono Lake. Now I know you are an expert already. When you see this food web, we're gonna look at again. It says here, review key organisms. You know this. You know who producers are. You know who consumers are. You know how to read them. We've been looking at all sorts of stuff with, and even in terms of when we look at the food web, trophic levels, that the numbers decrease as we go up. I know that you know all about energy flow. There's a lot of concepts when we do look at this little food web. And again, I know that you guys are pro with this. I know that you get this, but we want to start to look at a couple of details now. And now that you know so much, we can start to play what if. And remember, what if questions are the real scientific questions. Remember that this is just a basic food web, but we've been spending a lot of time recently talking about the pyramids, that our energy is greater down here, decreases as we go up. We know why, and our biomass, our amount of food, decreases as we go up. We know why. I want to start to look at one key relationship here. I want to start to look at just this little lower area, this relationship with planktonic algae and brine shrimp. And we're going to forget about all these other critters for a second. Now you might ask, well, why? Why would we do that? This is a really, 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 really crucial two sets of populations for this environment. Because if you look, the brine shrimp are a huge amount of food for a huge number of organisms, and the algae are actually what feed the brine shrimp, and actually even feed some others as well. So we're going to focus on these two populations today. Now, remember, a population is just a group of one kind of organism in an area. So we're in Mono Lake. So how many brine shrimp we have is in Mono Lake is the population of brine shrimp, obviously, and how many algae we have here as a population. And remember that when we say the population, the greater the number of brine shrimp there are, the more food there is, the more biomass, the more energy. So the numbers really do matter. And I know that we've been looking at the population pyramids, and that the numbers and biomass and energy decrease as we go up. So what we want to do today, there are scientists that did a study. They noticed something about Mono Lake. They kept going back throughout the year, and they looked at the populations and they tried to count them and see how many they are. And they noticed something that was kind of interesting. And it's probably true for a lot of ecosystems. Sometimes there were almost zero brine shrimp in the lake, like literally none. There were maybe like one or two, or in some cases, maybe none. They couldn't find any. And then at other times, there were literally trillions, thousands of billions. Just that number is staggering. It would literally account for there to be tons of them in the lake. So immediately, scientists were like, "Well, wait a minute. Why? Why is that?" So they guesstimated, or they reasoned. They said, "Wait a minute. There's got to be some reason why," and they called these reasons limiting factors. There's got to be some reason why the populations are small sometimes, and other times they're ginormous. They're huge. So they try to determine, well, what is it that's causing it? What are these limiting factors? So there's our focus, our essential question. What are the limiting factors that affect algae and brine shrimp populations? What are the reasons, really, why they're zero sometimes, and sometimes there's trillions of them, literally tons of them? So what they did, I'm going to show you. This is from our FOSS-Web book, and you'll have access to that. They set up a bunch of pretty easy experiments, and they did them right in a lab. They didn't have to go outside and do them. They did them right inside of like a laboratory or a classroom setting. So what they did, they set up a bunch of tanks, and I'm going to show you what these are. And they were able, and I'm going to show you the data as well. They did these tanks, and they changed in true scientific fashion. They changed some variables, some factors here. So What we want to do, this link is going that you'll be able to click will take you to this. These are literally the pages from our FOSWeb book. And they'll tell you all about these experiments. They set them up because they wanted to determine, they focused on two things here we're going to see. They wanted to know if the abiotic, the non-living factors of light and temperature, limited population growth. Like in other words, they're saying like, hey, if there's not enough light, is that going to mess with and change the populations? Or if it's too cold, or if it's too hot, or if we change the temperatures, is that going to affect the populations? So all they did, it's super simple. It looks confusing, but it's actually not. They took 4 different populations of algae and 4 populations of shrimp, and they put them into these controlled environments. Really, they're aquariums, we're going to see. They plopped them into aquariums where they could control everything. They made everything exactly the same in the aquariums, except for the one variable, the one factor they wanted to study. So here's what we said here. They set up 4 aquariums. They had the same amount of monolake water, the same amount of nutrients like CO2, the amount of salt that was in there, nitrogen, whatever, and a small population of algae. And 2 of the tanks they kept at a constant temperature, one at a low temperature, one at a high temperature, and they varied the light. So in here, they kept, for example, you can see they kept this tank really cold, 2 degrees Celsius. And everything else is the same as in this tank. This tank, they kept it warmer, 21 degrees Celsius. That's room temperature. That's pretty warm. And what they changed here, the amount of light. They varied it from a little bit of light, 9 hours, to 10 hours, 11 hours, 12 hours, 13 hours, 14 hours. So they varied, they changed the light to see what happened. And then in the other 2 tanks, they did the opposite. They kept the light exactly the same in each case. 9 hours here, and then they just varied the temperature over time. Sometimes they made it cold, sometimes they made it warmer, sometimes they made it really warm. And then in this tank, they kept the light really long, 14 hours. And what they did, they changed the temperature again. So we've got 4 different mini experiments for the algae here to try and see, is it the light that's going to change the populations, or is it the temperature, or what's going on here? Now, the genius thing, the great part, is that they did also with the brine shrimp. Remember, there's a relationship there. They got the 4 tanks. They put in, remember, everything exactly the same. They put in some algae, the food for the shrimp, and they put in 1 gram of eggs. And they did really the same thing. They varied the light, and they kept the temperature low, or in another case, another study, they kept it higher, but they changed the light. And then they did the same thing here. They kept the light constant at 9, but they changed the temperature. They kept the light constant at 14 hours a day, and they changed the temperature. And so then once you get them set up, all you got to do is let them run. This describes all of the stuff here, and you'll have access. You can go back and look at these. And remember, this modeled the conditions. It's a mini model. There's that word again, of just like Mono Lake over the course of a year. So again, remember, that is what they're after. So then they check the populations of shrimp and algae at the end of every month. And they counted the numbers of shrimp. They measured the amount of chlorophyll. That's a pigment that's in the algae, helps them do photosynthesis, tells you how much algae there is. And they did that over the course of a year. So at the end of this, they show you all the data, and they do it month by month, and they show you the variable. They color coded it. It's going to take you a second to look at this, but you can see right here, this is the algae population, how much in each month under each condition. So like, for instance, when we changed the light in the cold water, here's what they did. When we changed the light in the warm water, here's what they did. When we changed the temperature, but we kept the light at 9 hours, here's what happened. And when we changed the temperature, we kept it at 14 hours. Here's what happened. So you can see all the data for the algae and all the data for the shrimp. And what I'm going to ask you to do is to go back. You've watched the video here, to analyze this data. Take a look at the data. Look at what they found. You can go back and review the experiments. And then your exit ticket is going to be these 3 questions. All right, so what we're really after here in this whole thing, remember, we are trying to see, and again, there's also paper versions if you want to see those, but I like the colored versions better. I'm going to ask you to do that, analyze them all, and then answer the tickets. And remember, what we are after, we're trying to figure out what are the limiting factors that affect algae and population growth, because populations change. And remember, in a food web, if populations change, it's going to affect other organisms in there too. Can't forget that. Everything's connected. So good luck. Take a look at